In the final lecture of the quarter, we'll be concluding our discussion on kinetics and talking about reaction mechanisms, so multiple step reactions, how they occur, and also discussing catalysis. And so the title slide is kind of an overview of catalysis and the differences between kinetics and thermodynamics. Right? Kinetics is how fast something occurs, and it's all based on this activation energy. The lower that activation energy is, the faster this reaction would go from reactants to products. Thermodynamics is all about, the change in free energy between the reactants and the products. It doesn't care about, the transition state. Okay. So what catalysis, what catalysts do to speed up reactions is they make this barrier smaller so the reaction can go faster. So this is the last two sections in chapter 16. So one thing we didn't mention last time was that molecular structure does play a role in reaction rate. And it's not only about how many times molecules bump into each other or the energy that they're bumping into each other with, it's also about the orientation. And so there's another term here, technically, that capital A in the Arrhenius equation has this factor P, which is an orientation probability, and Z is the collision factor. So the orientation of a molecule will also play a role, right? Because sometimes, and this is an example, NO plus NO3, sometimes if they hit each other in a wrong orientation, no matter how hard or fast they're going, it's going to be ineffective, not yield a product, right? So they have to hit each other in the correct orientation for the reaction to occur. Right? Probably not something we're going to really consider in this class, but just to be complete, we'll include that. Right, so one theory that we're going to talk about is the transition state theory. Right? And that's that an effective collision between particles leads to the formation of a transition state, also known as an activated complex. Okay, the transition state is unstable because it contains partial bonds. Okay, it's kind of like a transition between reactants and products. So it's like a part-way species that's part-way between them. Transition states can't be isolated. You can't purify out a transition state because they are unstable. They exist as a point of maximum potential energy. And the energy required to form the transition state is thus the activation energy of the reaction. So example here gives bromine trimethyl plus OH minus goes to Br minus plus CH3OH. Right? So that transition state here, we'd have a bromine that's kind of connected to this methyl group that started on the bromine, and then it's also connected to the OH minus, and we show that via these dash lines. So this would be the transition state, showing the methyl moving from the bromine to the OH minus. Okay, and it's sort of halfway in between. And this, you can see that the molecular geometry this transition state makes, the carbon around the carbon, right, that steric number would be 5, so this would be like a trigonal bipyramidal shape around that carbon. Now, if we look at this in moments in time, right, if we have the bromine connected to the methyl group, and it's moving towards that OH minus, at a certain point, right, it comes to where there's these partial bonds start forming. This bond between the bromine and the carbon starts to decay a little bit, and this bond between the carbon and oxygen is starting to form. At the transition state, they're sort of, I guess you call it equidistant from each other. Then the carbon starts to get closer to the oxygen as we form that product. And finally, at the very end, we have that full product and the Br minus left over. Okay. The book shows this picture too with electron densities. I don't really like those as much, but what that's showing you is just how the bond, is starting on the carbon-bromine bond, and it starts forming in here as carbon-oxygen, and then finally the density is only between carbon and oxygen, and this bond is fully broken. We can do this too for some other molecules. Here we have 2 NOCLs going to 2 NO plus Cl2. So that transition state, right, the chlorines are going to, on 2 NOCLs, are going to face each other. And the transition state would look something like this. Here we have NO plus O3. Our transition state is going to look something like this. But in both of these cases, this is a potential energy curve, right? In one case, we have lower starting free energy values of the reactants, and in the other case, we have lower free energy of the product. So one would be an endothermic or endothermic or endogonic reaction. The other would be exothermic or exogonic. But in either case, they both have this activation energy, and they both would make a transition state. Also notice too that you have an activation energy for the forward reaction. So in this case, this would be the activation energy for the forward reaction. The activation energy for the reverse reaction is going from this starting point now, so it's higher. Okay, so first practice question. A key reaction in the upper atmosphere is ozone plus oxygen going to 2O2. The activation energy is 19 kilojoules of the forward reaction. And the delta H of the reaction for the reaction as written is negative 392 kilojoules. Draw a reaction energy diagram, predict a structure for the transition state, and calculate the activation energy of the reverse. Okay, so we have our reaction. Ozone plus oxygen goes to 2O2. So if we wanted to draw our diagram here, we know that if we're plotting now energy or delta H, we know that it's a negative value. So here to here, that's going to be delta H, and that equals negative 392 kilojoules. And we're also told that the activation energy is 19. So if we draw a straight line out to where we start for the reactants, we know that this value is our activation energy, and that equals 19 kilojoules. So what do we have? At this point here, we have reactants. We have O3 plus O2, plus O, not O2. And here we have 2 molecules of O2. So what is the transition state? Well, the transition state's going to look like an ozone coming together to meet with an oxygen. And so it's going to start to form an interaction there, and this interaction will start to break. So I really should have drawn that line as a dashed line for a transition state. So that would be our transition state if we draw just a basic picture. And how do we calculate the activation energy for the reverse reaction? Well, you can see from this diagram, the reverse activation energy is just going to be the activation energy of the forward plus the energy of the delta H. So since in this case it's a negative value, we would subtract delta H for the reaction because we're going in the opposite direction. So a negative of a negative will become a positive. So this would be 19 kilojoules minus negative 325 kilojoules. 325, what am I saying? 392. Which is 411 kilojoules. So far, we haven't really discussed how reactions occur, and that's what this term mechanism means. Okay, the mechanism of the reaction is the sequence of single steps that will make up an overall reaction. The individual steps of the reaction mechanism are called elementary steps because each one describes a single molecular event. So we're describing what happens on a molecular scale. The overall chemical reaction shows overall what's occurring chemically, but that doesn't necessarily show what's happening at the molecular scale. And that's what these reaction mechanisms, these steps, are showing. Okay, each step is characterized by its molecularity, and that is the number of particles involved in the reaction. And we'll explain that in a second. Rate law for an elementary step can be deduced from the reaction stoichiometry. So unlike in an overall chemical reaction, we always said, the rate law, you can't determine the rate law based on stoichiometry, right? Those exponents, you don't know them. You have to determine that experimentally. Well, if you have an elementary step, that elementary step you can think of as being determined experimentally, and it shows the exact amount, how the molecules are interacting with each other. And so that can be used to deduce the rate law. And in an elementary step, the reaction order equals the molecularity, but it's for those steps only. So if we look just generally, if we have a step that, and these are elementary steps, if you have A going to product, right, this is unimolecular, right, because you only have one molecule of A turning into a molecule of product. So that rate law for that elementary step would be K times A. If you have two A's now coming together to form a product, that would be what we call bimolecular. There's two molecules involved. And the rate law would be K times A squared, right? So now we can use that 2 in the elementary step, that coefficient, to be our exponent. The other type of elementary step that's bimolecular you could have would be A meeting up with a different molecule, B, to form a product, right? So that again involves two molecules, bimolecular. The rate for that is K times A times B. And then finally, probably the most complex thing we're going to consider is a termolecular reaction. So that would be 3 molecules coming together. So the example here is 2A plus B coming to product. That's termolecular. It needs 3 molecules reacting together. The rate for that one would be K times A squared times B. you don't see a lot of termolecular mechanisms because the chances of, three things coming together is much greater than the chances of two things, two molecules. And likewise, 4 or 5 molecules coming together is really, the chances of that happening is very, very small. So we just don't consider them in this class. Okay, so let's try a practice problem. The following elementary steps are proposed for a reaction mechanism. And these double arrows never come out, right? So this should be a double arrow. So we have 3 steps. Okay, write the balanced equation for the overall reaction, determine the molecularity of each step, and write the rate law for each step. Okay, so again, we have those arrows should be there. So to do an overall reaction, if these are our elementary steps, this is just like Hess's law from chapter 6. If something's on both sides of an arrow, we can cancel them out. And then we just add up everything that's left over. So if we look, Cl is going to be on both sides of the arrow. There's 1 Cl and here's 2 Cls. Those cancel. CCl3, both sides of the arrow. And I think that is it. So our overall reaction now becomes Cl2 gas plus CHCl3 gas goes to HCl plus CCl4. Right, so if we had just that overall reaction, we'd have no idea of these steps, and we'd have no idea that there were these intermediates occurring, the Cl and the CCl3. We'd have no idea that those species are actually present. Okay, but they're intermediates because they form and then they get used up. Right, so that would be our overall reaction. Now, if we look at the molecularity of each of these steps, so for step 1, we have a Cl2 molecule that decomposes into 2 Cls. So this would be a unimolecular. Right? Because we only have, in the direction it's written, we only have one molecule reacting in the reactants. Okay, step 2, don't want it to zoom in. Step 2, we have 1 and 2 molecules, so this is bimolecular. And step 3, again, we have Cl and CCl3, so that's bimolecular. And for part C, we need to write the rate laws of each of these steps. Right, so the first one, we have our rate is going to be equal to rate constant for step 1, K1. And the only thing it depends on is Cl2. Okay, for 2, our rate is going to be equal to K2, K sub 2. And now it's going to depend on the concentration of Cl times the concentration of CH2. CHCl3. Okay. And then finally, rate 3 will depend on Cl and CCl3. Okay, so pretty straightforward, pretty simple to work with these elementary mechanistic steps of a reaction. When it gets a little bit challenging, when we go into this in a little bit more detail, right? If we have multiple steps in a reaction, they're not all going to operate at the same rate. Okay, some of them are going to be slow, some of them are going to be fast relative to each other. The slowest step in a sequence of these reactions, the slowest step is going to have the biggest effect on the overall rate of the reaction. So we call this the rate determining or rate limiting step. So that's the slowest step in a multi-step mechanism. Okay. So for example, if we have the reaction NO2 plus CO going to NO plus CO2, this has been proposed to occur by a two-step mechanism. So the first step is NO2 plus NO2, so two NO2s reacting together to form NO3 and NO. The second step is NO3 plus CO going to NO2 plus CO2. Okay, so if you look at the individual rates of these two steps, the first step is the slow one, second step is fast. So the first one is going to be our rate determining step because it's slower. Okay? And the observed rate law for this overall reaction, you determine that experimentally, and it's determined to be K times NO2 squared. Right? And so if you're looking at just this overall reaction, right, you have no idea. You'd have no idea just by looking that this is the rate law, right? That would be determined experimentally. But the reason why it's like this is because you have this two-step process, right? So the first step involves two molecules of NO2, and that's why we have this dependence on NO2 concentration squared. And the rate law for that rate determining step becomes the overall rate law for the overall reaction, because the fast step doesn't really factor in. So how do you correlate the mechanism with the rate law? Well, a valid mechanism must meet three criteria. The elementary steps must add up to the overall balanced equation. So any steps you have in a reaction mechanism have to add up to that overall reaction. Okay, the elementary steps must be reasonable. I guess everyone's definition of that might be different. But having 6 molecules coming together at the same time is probably not very reasonable. That probably doesn't happen very often. So, right, they have to be in the realm of possibility. Right? And then finally, the mechanism must correlate with the observed rate law. So the rate law that's determined experimentally for the overall process has to correlate with what you would calculate based on your mechanism. Okay, a mechanism is a hypothesis. You can't really prove it is correct, but if it's consistent with the data and can be used to predict results accurately, it's then a useful model for that reaction. Okay, so we can never really prove these mechanisms. So if we write, let's look at a mechanism now that has a slow initial step. So our overall reaction is 2NO2 plus F2 goes to 2NO2F. Okay, the experimental rate law is K times NO2 times F. The accepted mechanism we have is a two-step process, so this is our overall reaction. We have 2 steps. Okay, the first is NO2 reacting with F2 to produce 1 molecule of our product plus an F. And then our second step is another molecule of NO2 then reacts with this F, this intermediate, to form our second molecule of product. Okay, so when you cancel the F's out that are intermediates on both sides of the arrow, this is our overall process. The first step is our slow step, and the second step is our fast step. Right, so our accepted or our rate law that's experimentally determined would be the same rate law that you calculate if you looked at just that first step, that first slow or rate limiting step, right? You'd have one molecule of NO and one molecule of F2. So the rate law would be determined, dependent on those two concentrations. Right, and again, we can check this. Elementary steps sum up to the overall balanced equation. Yes, both steps are bimolecular, therefore they are reasonable. And if we look at the rates of each, you see that the rate of 1 is equal to that overall experimentally determined rate law. And that is the slow step, so that correlates with the observed rate law. So this is a good mechanism for this reaction. Okay, you can also have a mechanism with a fast initial step. So if we look at this reaction, 2NO plus O2 goes to 2NO2. The experimental rate law for this is K times NO squared times O2. Okay, the proposed mechanism now is NO plus O2 goes to NO3, and NO3 plus NO goes to 2NO2. Right, so this second step is our slow rate determining step. Okay. The elementary steps do sum up to the overall balanced equation. Both steps are bimolecular and therefore reasonable. We don't have anything crazy going on. If we look at the rate for the first reaction in the forward direction, right, since this is fast, it means it's reversible. Okay, so rate in the forward direction would be K1 times NO times O2. We can write the rate in the reverse reaction as well as K minus 1, K sub minus 1, meaning it's going in the backwards direction. And that's only dependent on NO3. Okay, and then the rate of equation 2 is going to be NO3 times NO times our rate constant K2. Right, so when equilibrium has been established in the first reaction, right, that's fast and it will act to establish a equilibrium and then wait till this slower reaction to go and actually make the final products. Right? At equilibrium, rate 1, the forward rate equals rate 1 of the reverse reaction. So we can set these two equal to each other. Okay, why is that important? Well, if we want an overall rate law, okay, we're not going to have, if we tried to write the rate law of just this second step, which is our rate determining step, right, it's going to depend on this NO3. And the problem with NO3 is it's an intermediate, right? So it doesn't show up in our overall equation, our overall balanced chemical equation for this reaction. So we have no way, really, if you were doing this experimentally, you'd have no way of knowing it's there or measuring it. So we want to convert this concentration into a concentration of products and reactants, most typically of reactants that we can use. So if we set the rate law from the forward reaction of number 1 to the reverse reaction of number 1, we can use this to solve for this concentration of NO3 and plug those values back into here, and that will give us our overall rate law. Right, so by doing that, right, we solve for NO3, and it would be K1 over K minus 1 times NO times O2. We then would plug that in to our equation for rate 2 into this value, and it will be, there's a typo in the book slides, so this should be K minus 1 here. Okay. So we plug that in, and what do we get? Well, we'd get these K values would all kind of sum together to be a a constant on their own. We have two NOs, NO times NO, so that'd be NO squared times O2. And so that's what we see, right? So this K value is just a composite of these K values combined together. So let's try a problem with this. Okay, this will be our last problem here. Identify the intermediates in the mechanism. So we're considering the same reaction we used in practice problem 2. So we have to identify the intermediates, okay, and then part B, assuming that step 2 is the slow rate determining step, show that the mechanism is consistent with the observed rate law. And it gives the observed rate law here, K times Cl2 to the 1/2 times CHCl3. Okay, so let's remind ourselves what our reaction mechanism was. And we can also, let's show those arrows, right? Can do the overall reaction again. The overall reaction was Cl2 plus CHCl3 goes to HCl plus CCl4. So what are our intermediates? Well, intermediates are going to be those things that are on both sides of the arrow that cancel each other out. Right, so things like Cl and CCl3. They don't show up in the overall. They're only intermediates in these steps. So Cl and CCl3 would be our intermediates. Okay, so for part B, it's a little bit more difficult. Now we have to show that this is consistent with the observed rate law. And they give us the observed rate law. Let's write that down. Is rate equals K Cl2 to the 1/2 times CHCl3. Okay. Well, let's look at the rates that we would write for each of these individual elementary steps. So 1, 2, and 3. So rate 1 would be K1 times Cl2. It's a unimolecular reaction. And we can also say, at equilibrium, because that's a fast reaction shown, should probably write that in too. Right? Step 2 is slow. The others are considered fast, and one specifically is considered reversible. Okay, so that would be the same rate of the reverse, so K minus 1 times Cl squared. Okay, for rate 2, that's our rate limiting step. We'd write K2 times Cl times CHCl3. And so this is, might be tempted to say, oh, that's our rate limiting step. We can write our overall rate in terms of this, but again, this is including Cl, and that's an intermediate. So we'd like to write our overall rate law for our overall reaction without being in terms of an intermediate. Okay, so we want only in terms of reactants. Equals K3 times Cl times CCl3. Okay, so if we look at what we really want here, we want to be able to use this rate law because that's our rate limiting step. The problem is there's a concentration of an intermediate in there, so we need to get rid of that. So to do that, the easiest way is to look at equation, the rate law for the first step in this mechanism, because that's only dependent on the Cl concentration. And it relates the concentration of Cl to the concentration of Cl2, which is a reactant in our overall equation. So if we solve this for Cl, then we can plug that concentration, whatever that is equal to, into the concentration for Cl in the rate law for our rate limiting step. Cl2 squared. So solving that for Cl would be the square root of K1 over K minus 1 sorry, this is Cl, not Cl2. Okay. So we've solved this for Cl. Now we plug this value, what we have here, into this equation. So when we do that, now we have the rate of step 2 is equal to K2 times the square root of K1 over K minus 1. Sorry, that's probably hard to read. Times the square root of Cl2 concentration times concentration of CHCl3. Okay. So does this work? Is this overall rate consistent with the observed rate? Change my pen color here. Well, let's look. So this K2 times K1 times the square root of K1 over K minus 1, that is just, we can consider that as K. That's our new K value. It's a constant. The square root of Cl2 concentration is, the same as that. And we're multiplying by the concentration of CHCl3, and that is that. So yes, this mechanism is consistent with what we are observing for the rate law. So you can have cases of two-step reactions where you have the first step is the rate limiting step. So in that case, you'd have a higher hump here. So the activation energy of step 1 would take longer. That would be the rate limiting step. And then you have another step here. So that activation energy is much smaller. So this occurs quickly. All right, the opposite is if you would have a fast first step and then a slower second step. Right, so the activation energy of the second step is much higher than the activation energy of the first. Right, so how do we speed up reactions? And that's what the term catalysis means. It's something that speeds up a reaction. And catalysts are not part of the reaction. They're not being consumed by the reaction, so they're not being created or destroyed by the reaction, but they're working to speed up the reaction. So in general, a catalyst provides an alternate reaction pathway, right? And by doing so, it provides one that lowers the total activation energy. So it's just providing a different path so you can avoid that big activation energy barrier. The thing about catalysts is they speed up the forward reaction. They also speed up the reverse reaction, okay? They're just dropping that activation energy, and so that they do that in both directions. So they're going to speed up the forward and reverse reactions equally. Catalysts do not affect the thermodynamics, so they don't affect the delta G or the delta H or the entropy of the overall reaction or the overall yield. If we look at a catalyzed reaction here shown in green versus an uncatalyzed reaction shown in red, you'll see that what's going on? Well, we're taking this larger activation energy and we're making it smaller. And by doing so, we're speeding up that reaction. What we're not doing is we're not changing the potential energy of the reactants or the products, and thus we're not changing the delta H of the reaction or the delta G of the reaction. If you were to go backwards now, the backwards reaction, you'll see that it also speeds that up because the activation energy of the backwards reaction is normally this, uncatalyzed. But if we catalyze it now, it drops the activation energy. So again, it will speed up the backwards reaction, just like it speeds up the forward reaction. Right, and here we have just a figure from the book that shows this. So if we add a small amount of sodium bromide to a solution of hydrogen peroxide, what we see here is oxygen gas forms very quickly as the bromide anions catalyze the decomposition of hydrogen peroxide. Okay. The intermediate is formed bromine 2, turns the solution orange, but the solution again becomes colorless because the Br2 is going to be consumed in the final step of the mechanism. So bromine here is acting like the bromide ions, are acting like a catalyst, speeding up that decomposition of H2O2. Some other catalysts you can have, metal. Metals are used oftentimes to catalyze reactions. So in this example, we have H2 gas being absorbed onto a metal surface, and then hydrocarbons can come close by, like this ethene can come close by, and that catalyzes the reaction of ethene to ethane. So this is going to, the metal facilitates this H2 molecule to be kind of separated into two separate hydrogens and thus react with our ethene to make ethane. And this is known as a heterogeneous catalyst in that it's a different phase than the reaction mixture. So this is a gas phase reaction, but the catalyst itself is a solid metal. In biochemistry, we talk about catalysts, and we talk about catalysts in terms of enzymes. So there's a couple different models or theories about how enzymes work. One is the lock and key model, and that you have what are known as substrates. So those are your reactant molecules that will come into an enzyme. And what an enzyme is, it's a protein that has an active site in which it uses to bind a substrate or reactant, and then it catalyzes a reaction. So here we have our enzyme active site. The substrate or reactants come in and fit in there. So as the lock and key model suggests, this active site really fits those quite well. They're in there, and then the reaction can occur. So the fast step would be the substrates binding The slow part is the actual chemical reaction. That'd be a rate limiting step to form our product molecule. So that's the lock and key. So that in that model, you have a fixed shape of the active site that fits the substrates. Probably a better model is what's called the Induced-Fit. So in that model, your active site doesn't fit the substrates. When they bind, it actually changes the shape of the enzyme to fit and stabilize the transition state of your reaction, which then can lower the activation energy and produce a product. So I think that model is probably better. In this class, you're not going to be tested over the differences between the two models. It's probably enough for you to understand right now that an enzyme is just a type of catalyst. All right, and this slide's from my biochemistry class. What are enzymes? Well, enzymes are catalysts, and that means they increase the reaction rates without being used up. Some interesting parts that I wanted to mention here. The study of enzymatic processes is one of the oldest fields in biochemistry, and that's dating back to the late 1700s. And the study of enzymes has really dominated biochemistry in the past, and it continues to do so. And why is that? Well, because they catalyze reactions, enzymes were really probably the easiest biomolecule to isolate and actually try to study and try to use for making things because enzymes could catalyze reactions, it probably made them very easy to sort of identify and discover and isolate as one of the first bulk biomaterials to study. And why use, for industrial purposes, there's some good reasons to try to use biocatalysts over inorganic catalysts. And what are these? Well, you have greater reaction specificity with enzymes, right? And that avoids side products that you'd have to later purify out. You can use milder reaction conditions. So why our cells want to use enzymes as opposed to inorganic catalysts, right? We can use milder reaction conditions, and that's conductive to the conditions in cells and the conditions life can occur at. Some inorganic catalysts, you have to heat up to a very, very high temperature to get the reaction to actually speed up the reaction, or be at high pressures. You can get very high reaction rates. So these reactions that might not occur very quickly can now occur in a biologically useful time frame. And then you also have the capacity there on enzymes for regulation. So you can be able to turn on and off an enzyme, whereas maybe an inorganic catalyst, you couldn't do that. Right, so if we plotted an amount of product produced over time, if we don't have an enzyme, you just kind of go up linearly. With an enzyme, you can produce a lot of product very quickly. It speeds up that reaction. And here are some rate constants for certain reactions that occur in cells. And so the uncatalyzed rate is shown here, and the catalyzed rate constants are shown here. Okay. So, and then the non-enzymatic half-life is also shown. So this reaction that the OMP decarboxylase catalyze, the non-enzymatic half-life for that is 78 million years. Okay? And the uncatalyzed rate there is shown as 10 to the negative 16th, so very, very slow. Well, the catalyzed rate now becomes 39. Okay, so that's not the biggest number, but compared to what it was here, that's a huge improvement. So the enhancement of that is something like 10 to the 17th. So some of these reaction enzymes can speed up a reaction very, very quickly. Another important reaction that gets catalyzed is the breakdown of ozone. So in the upper atmosphere, ozone, that ozone layer is very protective. It can be broken down by chlorine atoms. So the uncatalyzed reaction, ozone plus oxygen, just a single oxygen atom, that has a fairly large activation energy. I think we mentioned it before as 19 kilojoules. In this diagram, it's showing as 17 kilojoules. But whichever it is, it's a very high barrier. Chlorine, and specifically Cl dot, Cl minus, can catalyze this reaction by creating this transition state or intermediate here, ClO dot. Okay? And that lowers that activation energy to 2 kilojoules. So you're going from 17 kilojoules to 2 kilojoules. So that speeds this reaction up a very great deal. Okay? And why that's important? Well, if you have a lot of these chlorine molecules in the air, it's going to deplete the ozone. Right? And if you look, we have the total ozone is given by this color chart. And over Antarctica, you see that in 1979, it was starting to, you're starting to lose some over here. I think naturally it's probably just a little bit lower on the poles. But by September 2008, because of use of these ozone depleting chemicals that get in the air, you can see that it's a huge difference, right? Your ozone is really completely depleted here. I think honestly, it has gone back up since we've kind of outlawed the use of a lot of these ozone depleting materials, so that's obviously a good thing. But that's just an example of how you can see catalysis working in an environmental chemistry situation. All right, so that concludes our lectures for this quarter. I hope you have enjoyed it as much as I have. Just remember the final, you can use a sheet, front and back of notes, just a normal size sheet of paper, 8.5 by 11, nothing crazy like a poster size. So just be aware of that. Obviously, if you didn't have that on the exam, it's going to be a little bit more difficult. All right. So good luck on the final, and have a good spring break.